Yeah, so that work, I actually was an undergrad at Northwestern, and I thought, I got involved in neural network modeling. I said, understanding how the brain works as a computer seemed like the most exciting thing. And so I encouraged, if undergrads in the room, that's kind of what launched me into all of this. I found a faculty member to work with, and we were actually, this was the 80s, right? So we were building backprop models, and biologically implemented gradient descent was what I was working on. But so now that's come back almost 20 some years later. But that's what launched me into saying, well, I'm not sure I want to be a doctor. Maybe this research thing will actually be quite exciting. So even though I did an MD, PhD, I don't practice medicine. I just do the PhD part. So what I'm going to talk about today is around what I'm going to call broadly science of natural intelligence, and that's partly because I'm advertising for our Center for Brains, Minds, and Machines at MIT. If those of you are interested in the intersection of cognitive science, neuroscience, and computational science, I encourage you to come as grad students or postdocs or hopefully faculty, because that's a big part of what we're trying to do there in the department. But my lab works primarily on broadly visual perception, and more specifically visual object recognition, as I'll show you. And we try to take what I call a reverse engineering approach, and I'll try to lay that out for you so you can get a better feel for what I mean. And I think this relates to Conrad's talk on understanding. I should kind of, by way, this is wearing my department head hat, that the mission at MIT of our department is to understand ultimately, and I use the word understand, how the brain gives rise to the mind. And to us, again, that means reverse engineering the mind and ultimately building models to link across all these different levels, as we discussed in the last talk. One person's mechanism is another person's phenomenology. My lab works up here at the level between systems of neurons that live in brain regions that are executing algorithms to support cognition. I'm not going to talk about anything down here at the cell molecular or smaller neural circuit or gene level. I also put this slide up because, because Conrad's talk reminded me of I like to talk about this in the department, that we think the brain is some sort of computing device, and this is a computing device. Conrad showed you a 70s computing device. This is kind of the structure of most of our modern computing devices, and that there's many levels of abstraction, physical abstraction, in which you could study such a device, just as I'm showing you here. Of course, these are not one-to-one, but I hope you get the idea. But what I want to point out is that it's not like a computing device that we've yet built. It's different in some ways. Conrad said it must be definitely more complex. You could argue it could be less complex. We don't really know yet. It's like in a device we haven't built, but we have an opportunity to study it. So to me, I think of the brain as a device. It's like we've been given from the future or from an alien planet, if you like. Wow, look at this amazing thing. Let's figure out its secrets, and that would allow us to do amazing things that we're not able to yet do. And here it is. That's the human brain. And it's remarkable that it's actually only 3 pounds and 20 watts. So just 20 watts in this amazing computing power. So that's our mission, is to try to understand that, to kind of extract its secrets. And it's, as I said, my lab works on visual perception. And I like to point out that humans have broadly what you might call strong perception. And this is what I've heard some people refer to this way. And what we mean by that is that you don't just sort of say there's a car out here, as I'm going to tell you about later in the talk, but you might be able to predict like its pose and what's going to happen next and what this person's going to do. You have a lot more inferences that you bring to this scene than just naming the objects and their positions in this scene, although that's what I'm going to talk about today. What we've been trying to focus on since 2002 when my lab started is to not reverse engineer full strong perception. That was too big of a problem. Let's focus on something simpler, which was object detection and categorization. And that would be, for instance, as I just said, being able to name the nouns in this scene here, and perhaps some of their other latent variables, like not just their identity and category, but say the pose of this car or its position in the scene. Now, we wanted to take constraints from biology. We don't say, well, you may not realize this, but you don't actually digest that scene all at once. You may think you know what's going on. Marissa had the nice slide where you had the change going on. You don't actually notice most of what's going on. One reason you don't notice is because you're usually using what's overt attention rather than covert attention. So when you're actually looking, when I put that slide up, what you were doing is your gaze might have fallen here. You have high acuity retina taking in largely the central 10 degrees. Of course, you can see, quote, see everywhere, but you have very high acuity in the central part of your field. That's the part of the field that's actually flowing down the ventral visual stream that I'm going to tell you about in a minute. So roughly, I think of the central 10 degrees, which is like 2 hands at arm's length, if you want to think about what central 10 degrees are. So if you were there, you didn't just sort of stay right there and say, analyze that intently, but you were using, again, overt attention to make rapid movements around this scene. So you're moving your eyes very quickly in saccades, as you know, as you probably should know, you don't see during saccades. You only see when you stop your eyes. So right here, and then you make a rapid movement, and then you're here, and then you make all these movements to sample the scene. And those are, of course, directed in interesting ways, and others study that. I'm not going to talk about the direction. What I want to tell you today is that now I'm going to reduce this whole problem of kind of understanding the scene to a much simpler problem that looks like this. I'm going to take out those snapshots from those samples at 10 degrees, and this is kind of what you get. And I hope that I'm going to show you this now a little bit out of context and see that you can still see a lot of things in the time scale of your visual system here. So I hope that you can notice, I didn't really play any tricks on you. You may be thinking I'm playing a trick. Did you see there was a car, there was a person, there was a street sign. You could pick up one or more objects and stuff about like their position and pose, even in those brief glimpses. I hope you guys all saw that. Even that kind of, you know the context because of the street scene, but I removed it. And this connects to older work. I refer to this phenomenon here as core object perception because I think it's core to sort of building up a larger understanding. Again, I'm operationally defining that as central 10 degrees and just 200 milliseconds of viewing. This connects to older work from Molly Potter and others way back in the 70s that showed this is called rapid serial visual presentation. I'm sure you've all seen this. The point, look at this, is like completely out of context. You can recognize, again, one or more objects in these images, even though they come by quite rapidly. This is a very old movie we got made as a postdoc, and I still like to show it. The point is, look, I didn't have to pre-cue you. I didn't have to kind of give you an attentional cue of you're going to see Star Wars characters or where they're going to be. You do a lot of power on just those central, that snapshot scene of just processing that immediately, and that's what we've been focused on. So I'm calling that core object perception. And we focused on that because that was a hard problem for computer vision. You know, we don't need to even move the eyes or to invoke covert attention or other things that we know happen in the visual system to say there's already a very interesting engineering problem to try to understand. So that's what my lab's really been focused on for the last almost 15 years now. And so I'll give you kind of a summary of what we've done, but just again to give you some idea, you know, this might be kind of how we test humans. This is like we might show them images, again, 8 degrees center of gaze, 100 millisecond viewing time, which is about the duration of your saccadic snapshot. It's about a little bit less, but it's similar behaviorally as I'll show you, and you can kind of try this for yourself. Again, I'm not even going to tell you this is like an 8-way categorization task that might run humans on, but here I'll give you a quick image. Anybody see anything? What did you see? Car, right? Okay, let's try another one. That's a hard one. Try it again. Airplane, yeah, so. Right, so they're completely out of context, yet you kind of like just go right through it and it's like instantly do it. You know, this is an 8-way categorization task and we ran this. Here's human performance on this thing. This is stimulus duration. That was 100 milliseconds roughly. And again, all the data I'm going to show you today is 100. This typical fixation time of a primate during active search might be about 200 milliseconds. Here's accuracy with those kind of durations of viewing. Again, knowing this is accuracy in an 8-way task sorted so that chance is 50%. Forget the details for now. All I want you to know is this is very high performance. You still can get higher performance if I give you longer time to look at it, but you get a lot of the juice out of the performance of this system on these kind of tasks in just a couple hundred milliseconds, and that's all I want to convince you of. So I'm going to call this the core object perception regime, and that's what I'm going to talk about today. So now I'm trying to position core visual object perception as kind of in the larger context of strong scene understanding, which is more than just vision. And also, I started with like natural intelligence, which is a very big thing. But just to say, this is the chunk that we're working on. We'd like to have models of how the system works within this limited domain as a building block to this larger set of things. Okay, now I kept using the word reverse engineering, and I'm going to try to say a bit more of what I mean by that. So I've defined a domain of problem, at least roughly operationally defined it for you, core visual object perception, naming nouns and those kind of durations and scene and image sizes. And so now we have to ask, if you want to do reverse engineering, the way I think about it is you have to do 3 things. The first thing you want to do is, why are we even bothering to do this? Well, you want to look at where does the engineered systems, the currently engineered systems, fall down relative to humans. So as I introduced this to you, I said, look, the brain is a really powerful thing. That implied it's more powerful than some currently engineered systems. So we were very interested in studying the brain, finding those reverse engineered secrets in domains where it was actually outperforming currently human engineered systems. And then you want to say, well, look, if I'm going to extract those secrets, as I said there, you've got to go in and measure some stuff. Now, it could be anatomy, it could be physiology, it could be blood flow. You can measure all kinds of things about the brain, but you have to think about what might I measure that might inform on that. And when you think about that, you have to think about, well, can I actually get such measurements? So this is kind of choosing what kind of experiments you want to do and hoping that they're going to constrain your understanding. And then this is often where neuroscience stops, and this is where I think I resonate with what Conrad said. Often then an abstract will get written, the brain is complex, we regress some things. But really then you have to do this step, which is you've got to do some serious forward engineering. You say, given these are just constraint data, you have to build models that are really forward engineering guesses as to how the system's working. And then if those models match to the data, you start to say, well, this is a description, a predictive description of the data. And to me, this is what understanding is going to look like. They're models that fit all the data that we have, both behavioral, neural, whatever levels that we say we want to describe the system, and they should generalize in their predictive power. And if you can build such a thing, then I would say that you do have an understanding. And I hope we can discuss this further because it relates to what Conrad told you. Okay, where do systems fall down relative to humans? Well, when we started this work, it was clear that what artificial vision systems were struggling with is being able to recognize objects in a way that's tolerant to what's called identity-preserving image variation. So for instance, this car can present an infinite number of images to your retina due to changes in viewing angle, like position, size, and pose, to the fact that you might say, look, I want to do car categorization, so now there's sort of shape level variation within the category, to the kind of obvious real world stuff that you have to deal with, which I showed you in simulation. This is a rendered car on a complex background, which is kind of what I was showing you earlier. You have background clutter, you could have potentially occluding clutter and lighting changes and so forth, and these are real world images from like computer vision tests. But I hope you get the idea. You can see these are quite similar in their sort of difficulty, yet each of us looks at those and instantly says, as we did earlier, to some high degree that there's a car in this image. And machines were struggling with that. We could show that here. This was back in 2009. This is if you kind of knew that the car would be side-facing, for instance, and right in the center of the screen. So this is called low uncertainty in object view. This would be high uncertainty, which is kind of like the set of images I was showing you on the last slide. I'll show you some examples of that in a minute. This is performance in D prime unit. Marissa said this in her talk. The higher you are here, the better you are. And human level, this is human data from a bunch of humans on doing the kind of thing that you were doing a minute ago. And you can see they're quite good. It's not as if they're infinitely perfect. So I like to say they're tolerant to variation, but they're not invariant. They don't have full invariant recognition. And so they fall off a little bit. But you see, what's most interesting here is that current machine vision models at the time, and this was, we call this a V1-like model. It was actually state-of-the-art in competitions on Caltech 101 at the time when we were building these things. And there's a whole backstory behind that about how computer vision progressed. But the point I want to make here is that in these low variations, these machines were matching us or could even beat us. But at these high variation conditions, humans were consistently outperforming machines. And we knew this. This is what we thought, and this is just a quantification of that and us trying one forward engineered system at the time to convince ourselves we were actually onto an interesting problem. Okay, so that was the domain that we were trying to study. And this is now, of course, now trying to say, well, question 2, as I said, let's go get some data from the system of interest to say, can we constrain it? And so we would love to. I've been talking about reverse engineering the human mind or the human brain. Of course, that's our goal. That's what all of us, I think, maybe in this room would love to do. But that system doesn't allow the access that we thought we wanted. And so instead of studying that system, we study this system. This is the non-human primate brain. It's not shown to scale here. But the reason we study the non-human primate is that we know from work of lots of people that have gone before us a lot about the layout of the primate brain systems. We know about the regions of the brain called the ventral visual stream, where if you have lesions in this large area here, you tend to get disruptions in visual recognition. Now, that doesn't mean it's fully sufficient in Conrad's language, but it says, look, something's important's going on in here, right? So this was kind of, we had some rough roadmap of where to look. We thought this is where the critical computations are likely taking place, not only from lesion data, but from other physiology data. That we had a reasoned guess that this is probably where interesting computation is happening in this system. And so we also know, just to orient you, the ventral visual stream, I'll show you in a minute, is a series of visual areas, V1, V2, V4, and IT, which is actually, we think of it as 3 different areas, shown here as 1. But IT broadly projects to the frontal parts of the brain involved in decision and action, and to other parts of the brain in the medial temporal lobe involved in the laying down of long-term memory. And so we think of these kind of neural population patterns, or what I call neural representations, but just neural activity patterns, as the substrate for doing these other kind of things. And hopefully that'll become clear in a minute. So we know about the system anatomy at coarse grain of this system. And we also have models of the elemental computations, again from work that had gone before us about how V1 neurons kind of compute on their inputs. And I'll show you some of that a bit later. So again, we knew a lot. We weren't coming into this blind about what a primate visual system sort of does. And because these are, as I said earlier, instead of this system, these are non-human primates, we can much more systematically measure and manipulate every stage of processing along the ventral stream at the level of spiking activity, at millisecond resolution, and in behaving subjects. And that's not currently possible in the human brain. Okay, so that sounds like you might have access, but you might have said, well, wait a minute, now you've given up the human, what about the behavior? Well, so here's monkey behavior. This is the other reason we like this setup, is this is a non-human primate. This is in his home cage. He's like, you know, getting a nice juice treat from doing this. They like to do this all day long, where they kind of, well, not all day, but many hours a day, they'll come in and sort of, instead of just sitting around in a cage, they'll come and play this video game where they press a screen, an image comes up, and they're trying to identify, you see him making a choice of 2 possible objects. And so this monkey knows something like 30 objects, and the green is meaning he got it right. You mostly see he's getting green. That one was wrong. Okay, stop for a second. But you get the idea of what these guys are doing, and they can collect a lot. We can collect a lot of behavioral data this way. We can get a lot of behavioral data, of course, from humans. That's a very good data source. And the upshot of all of this is when you compare humans and monkeys in their behavioral abilities, they're very, very similar. And I don't want to bore you with the details of this slide, other than what you should see is like these look very similar. And what these are kind of confusion patterns of, say, how difficult it is to discriminate a truck from a tank. And red is high level of confusion, and green is, or blue is low levels of confusion. And all I want you to see is they make similar patterns of difficulty across, this is what we call the object grain. It's averaging over many images within category. But the upshot is, again, these are very, very similar in terms of measured behavioral difficulty. These patterns look very, very similar, which to us implies some sort of shared neural substrate or bottleneck, shared neural representation that limits the behavioral performance here. And so this shows you that this is a system, if you're interested in this task of core object categorization, that this is a good system in a very quantitative sense, not just that we might get some principles of computation, but actually this is almost a copy model of what we might have in the human system for these kind of tasks. Okay, and we have access, right? So now let's use that access. So we have this monkey ventral visual stream. We think of the ventral visual stream as engineers as being, there's millions of neurons in each area. I'm schematically showing you them here as these sheets of neurons here. We have forward flow of information through anatomical connections this way, intercortical connections, and then feedback connections here. So you have this is just a schematic of just the cortical areas. I'm not showing you the subcortical areas of kind of the feed-forward feedback flow down the ventral visual stream. And then the kind of rough lay of the land that we knew when we started this work is that an image comes in, the retina makes a nice photographic copy of it, processes it in nice ways, transmits it rapidly through the LGN and the thalamus and through this series of visual areas with about 10 millisecond lag on average between areas to about 100 millisecond response patterns in IT. So what I'm showing you here, each dot is meant to be the activation of a schematized individual neuron. Roughly 10% of neurons are activated at each level by an image flash such as this one, so that you end up with a pattern of neural population activity up here that started as a photograph and now looks like some pattern of neural activity that's actually nothing like a photograph anymore, but turns out to be quite powerful, and it's produced in only 100 milliseconds. New image, new pattern. When you keep doing this RSVP task, your homolog of IT is clicking along at a lag of about 100 milliseconds following along. Okay, so you have this kind of, now this setup of the system that's computing what we think is interesting information about something about the objects in the world, but our task was to try to understand it. And again, in around 2000 when we started, we had very little engineering spec, very little in the way of models that could actually explain what was going on beyond these early levels at these kind of population level image transformations along this pathway. And perhaps even more importantly, even though I've been implying these are important areas, we knew that these parts were kind of important with you lesion them, but we didn't have any model, a quantitative model, a predictive model that would take us from this neural activity to actually explaining the behavior that I set up for you, the core object perception. So those are still the 2 core questions in my lab, and we made a lot of progress in both of them, and I'm going to try to take you through some of each of them now. I should point out, and maybe this is already said, is that because we're working on non-human primates, we can do not just behavior, we can do neural data collection recording that I'll show you in a minute. We can do things like fMRI and other imaging techniques. We can perturb optically or pharmacologically, and of course, we build models to try to understand what's going on. So these are the kind of tools that we most use within the lab. Okay, so we didn't know much. Again, now I'm going to turn to this problem about how you relate neural activity to behavior. I've been implying that that's important, but I'm going to show you a bit of why we think that. So back in 2005, working with Tommy Poggio and colleagues at MIT, we just did this simple thing where we recorded from neurons. These are simple images here, and this is just to give you a feel for what neural spiking looks like. Conrad showed you some simulated spiking. These are 3 different IT recording sites. Each tick mark is an action potential. And all I want you to see is the different sites. You put on an image that's on for 100 milliseconds here. You have a lag, again, about 100 milliseconds. You see this increase in firing rate. And you see that there's some selectivity across these images. High, high, medium, kind of low, if you sort of integrate those spikes with your eye. And you see this site in low, high, medium, sort of medium. And you see there's different patterns of selectivity there if you kind of average across those spikes. And that's kind of what we're going to do next. We're going to ignore all the individual spike timing details and just give you this sort of average response over a time window. Because what we found is that when you look at those kind of spike patterns within IT, these population patterns, that they were very powerful for supporting this core recognition task, much more powerful than machines were at the time. It's as if we kind of found or tapped into a solution output internally of the brain to something that was hard for computer vision at the time. So let me take you through how that works. And again, this started in 2005. It's still the same basic decoding model, we call it today, that we use today, which is you take the spikes that you might record out of a population. I don't know if you guys can see that, if we can turn down the lights. This is a population of n neurons. And n, we can vary n from any number of neurons that we collect. Oh, thank you. This would be the response to one image. Here's a bunch of spikes that come out. These are just simulated spikes. We can count spikes in a window. This would be the simple average count within a 100 millisecond window. We call it a 100 millisecond rate code. This is something that we're of great interest to us, is like, what are these proper counting windows? But I'll just show you this simple one for now. And you can see that that transforms the spiking data on each trial to a kind of response value from each neuron at this time window. So neuron 1 would have this value, neuron 2 this value. So now you have a set of n neurons, you have n values, so it's an n-dimensional vector. You can think of it as a point in an n-dimensional space where n is the number of neurons, and this would be the response to this image. Maybe there's trial-to-trial noise that is schematizing there. And the basic idea that we were after and that we tested is, look, you're going to have many images of faces that you want to discriminate from non-faces. So faces in green, non-faces in red. And to discriminate means that we imagine that the information is such that with a little bit of labeled data, such as someone saying, oh, that's a face, that's not a face, some labels that you could separate the data with simple linear tools, which are basically, these are linear classifiers. Just think of a hyperplane in this high-dimensional space, a plane trying to cut the data. And what that amount, the reason we use that is not just because it's a nice tool in machine learning, but because it's an approximation. If you think about neurons as weighted sums with thresholds, that's essentially what this hyperplane is doing. So we considered kind of hyperplane decision making on neural population space as approximating what might be going on downstream. So when you build those kind of decoders on these neural populations, the upshot is they're very, very powerful at predicting the animal's difficulty with tasks and really just being able to do the task at all, to say, hey, this is a face in a way that generalizes across these kind of image variation level. Again, better than machines at the time. So I'm telling you that without showing you too much data, but I'm just going to try to give you a feel, again, for how we did it. We had image tasks that look like these. These would be kind of images that we would show. This is more recent work. Face versus not face, that's a discrimination boundary. You collect a bunch of neurons, you train up a classifier with some images that you say, these are learning examples, and then you test performance on held out examples such as this. This would be one task for us. Here's another task, beetle, not beetle, this is a type of car. Again, you can see high variation, random backgrounds, all that makes it difficult for machines, yet humans, again, and monkeys still do quite well. Then you say train up on some, test on some others. And this is just to give you a feel for how we actually do these studies. And I'm saying all this again by saying, look, you're recording from IT, you're collecting population data, and then what are you doing from a neuroscience point of view? So the way we think about what we're testing is the hypothesis that there's a feature set produced in IT. And this is not a new hypothesis, we're just implementing it. That there's a feature set produced in IT, there's some downstream read of it that has to get trained up to say, hey, this is a car, this is a dog, this is a cat. And that is the mechanism by which you can support future categorization performance on these images. And just to give you a feel in practice operationally what this means, this is what monkeys look like training to learn a new object. So what's great about monkeys, and this came up in the first talk, is they don't know what a car is when they walk in because they haven't been asked to drive or anything, but they can easily be trained to say that's a car image and that's not a car image. It takes them about a day to learn a new object like that in a way that's near human level, near or just at human level performance. This is tests over many objects. That's average learning rate. Ignore all the colored dots, but this is many, many objects. As monkeys learn about one object a day when you do this. And then once they do this, they're at human level performance, and they have the same patterns of difficulties as humans. So they have to be trained up a little bit over some time window. So back to the brain here, what we think is going on is when that monkey is learning that, we don't think its whole ventral stream and its IT are changing around, and we have some evidence to support that. Somewhere downstream of IT, it's learning to map that feature set onto an appropriate behavioral output. And where that is actually an open question in the lab. We have ideas. We think this is in the caudate for some of the tests, and this is an area that we're working on now. But I'm just putting this up so that you can say how we conceptualize what we're doing and how we think about it in the context of the brain. An IT powerful but relatively fixed feature set, followed by rapid learning with some training examples to kind of get the monkey to do these kind of behaviors. That's our kind of working model of what's going on. And the further we test that model, it just keeps holding up. We've been able to collect more and more data with array recordings that are chronically implanted so we can get large volumes of data. When we first did this in 2005, we had much less data. Now we can get a whole lot of data out of these arrays because we record from the same sites every day. And I'll just quickly take you through this. You get, take spikes from one site. You can take the mean response. This is one site to 10 images of one particular category called animals. You can get the mean response to these images shown here, just like I showed you earlier. Here's a bunch of categories, and then you take the mean, and then we actually now had a bunch of images of these, and this is just one site of hundreds of sites that we're recording. So you start to get a feel for that we can get these large volumes of data. And really, the way you should think about these kind of data is you end up with some number of IT neurons, which ranges from 100 to 1,000 or so, and that's the response vector to one image. Maybe 8 images, we actually collect 50 repetitions of thousands of images. So we end up with this very large data matrix and at high SNR, and this is what we've been using to do a lot of the things that I'm going to show you about in the last part of the talk here. So the upshot of everything that I'm trying to introduce you, and this is background for me, I'm just hoping if people haven't seen this to bring you up to speed of how we think about the problem, is that by collecting a lot of this behavioral data that I showed you and these kind of population recordings out of IT, what my lab and others have helped to build up is this basic idea that the IT feature set conveys what I'll call biology's solution to this initial object intelligence sensing problem. That is what's out there and also where it is in its pose, which I'm not going to show you today, but it does that as well. And it's rapidly computed. In just less than 200 milliseconds, this high-performance feature set comes out in IT cortex, as I conveyed earlier. It's a general basis. So as I said, you can learn new categories on it. You don't have to change the basis. Even though the monkey didn't know a car, it had a basis set that could quickly learn what a car is with just a little bit of training. And that's consistent with the way machine learning folks think about a basis set. You want a good feature representation, and then a little bit of learning is all you need to do well. And it also supports, as I said, object position, pose, scale, et cetera. Beyond just saying different objects, it supports multiple tasks, task types. And what I really hope that we've contributed here so far, that idea that I've been putting out there has been out there for a while about a feature set in IT, and you can learn from it. But we're actually building quantitative models that are now predictive in a quantitative sense of how difficult every task should be. If we perturb the neurons optically, which we're doing, how much behavioral effect we should have. So we have kind of what I call an engineering spec model of all of the above. And just to give you a feel for that, this is kind of predictions, sort of actual behavioral performance, predicted behavioral performance. This was a few years ago now, but this is just to give you a feel that not all tasks are equally easy for an animal or a human, and the model predicts that actually within the limits of the noise that we had at the time that we did this study. So that is just to give you, all I'd like you to take from that, and I'm going to pause for a minute here, is that IT feature set conveys a solution to intelligence sensing. It's like we feel like we've tapped into a solution. We understand the form of the solution, at least something that seems to fit the data that we have so far. So before I go on, I want to kind of stop here and see if people have questions about that, because I know I tend to go fast. So anybody have any questions about that so far? Yeah. So there are many studies that I'm kind of all lumping together when I'm talking. So when you say the data recorded from IT, So if we just do like this one, for instance, or this one. That is data from, these are data from, I think about 168 IT neurons. This is about 5 years ago, and I just haven't gotten a nicer slide. It's about 168 IT neurons from the same, this is actually, sorry, pooled across 2 animals. These were the first 2 animals we recorded with this array method for a couple thousand images. So this is 168, and those are each color is the mean response averaged over 50 repetitions of an image. The mean, like think of that 100 millisecond window, 50 reps of that gets you one number averaged over all of that. That's what the green color is. So there's 168 here, and this is like 2,000 images across here. The 8 lines are like 8 different categories that we had where the images are lumped into categories on there. That's what this is. Does that help? That's kind of the data. It's like thousands of images. We kind of group it in categories, but there are many images per category. Okay? And the images you saw, they were kind of rendered objects on complex backgrounds, although we've collected other stuff more recently. But that's mostly that's what this is here. Any more questions about the kind of setup here? Yeah, yeah.
When you talk about IT feature sets, can you say more what you think the features are that are feeding into these categorizations?
There are features that support categorization. So I think this bears on Conrad's talk about, like, I think you're asking me for tuning curve description. And I just hesitant to offer that. I think that'll come up again next. I'll show you some of the responses. You can tell me what you think they are, right? Or I could say they're the outputs of a deep neural network that I'll also show you next. And both of those are descriptions of what we think they are. Okay, so that's how I would describe it. So I saw one more hand, or did I? Yes, or 2 more, yeah. No, this is good. I want to make sure you guys are up to speed, because this is for you, not me. So yeah, go ahead. Yeah, I think you're asking, like, if we don't know exactly where each of these IT neurons are projecting. Right? We know they're roughly projecting to the areas I showed you and others, but these are a random sample out of IT. Right? And I think this is a great question, right? And then it's sort of interesting that we can take a random sample and actually fit the behavior quite well. And I kind of showed you that with a slide where I had that IT, and I said, here's these random connections up to a neuron. You learn, and it kind of prunes the connections, right? And that's my preferred hypothesis, because it's like, well, that's where our lamppost is in the way that we're looking there. And so I'm kind of admitting that I don't know, that a more detailed knowledge of the anatomy wouldn't give us more insight. I can't say that. But I also think, look, when you're actually building a brain, when the brain's developing, it kind of has to struggle with the same problem that we're struggling with. Like, some wires come at me, some neurons are and I have a local learning rule that I have. And so that's why I think this is not outside the realm of possibility. And then what you might start asking is, do the time rates of the learning that you see and match the time rates that the animal does? And those are the kind of studies we're doing now. Because that's the one way we can get at that question. But it's a great question, right? I mean, and we could talk about face patches and kind of what are thought to be known structures. Those are functional structures. They're not anatomical structures, really. But we could talk more in questions. But hopefully that's enough for now. Yeah. Maybe one more, and then we should go on. Yeah. That's what we did in this example, but we usually vary that and try all kinds of things. But well, an integrated code is kind of a low-pass filter. I mean, but we've done smaller time windows and bigger ones, and so far a lot of those codes can still fit the data we had, but we have higher res. What you need is high-res behavioral data to really tease apart these alternatives, and we have that now. We didn't have it at the time we did that study. I'm not ready to tell you what those answers are, but we think there'll be some more traction there on the temporal code. It kind of comes up right at the end of my talk. Maybe this will come back again. I mean, that's another great question. Is like, just our first model is kind of approximately right. This has come up in both these questions, but I'm sure it's not exactly right. It's just kind of our current best guess. And so these are ways to refine our understanding. And these are exactly the right questions and the things that we're trying to do in the lab. So I don't want you to think we've closed the door on this is exactly how IT gives rise to the behavior, but I just want you to see it actually works pretty well. And it worked as well as we could measure when we did it. So, but we're trying to measure it harder to see if we can break it. That's what a good experimentalist should do, is like beat up on your theory. Okay, so let's go on to the second part of the talk, really, which is, how much time do I have? How am I doing? Okay, so in 2000, we had almost no spec of what was going on along the stream here. I said that earlier, but so now let's do this forward engineering part. We already did a little bit of forward engineering on the decoding model, but now we're going to try to do some more forward engineering here. And remember, it's not like we knew nothing about the layout. I've already shown you a lot of stuff here, right? So we knew a lot from before our lab even started. Hey, there's a, quote, deep stack of areas. You know, I already drew that for you. This is, you know, decades old. Each area conveys a retinotopic map. So there's, you guys probably know this from your intro classes. There's a retinotopic map all the way down here, not so much in IT, but pretty much everywhere else. That each area, you don't, when you take a V1 neuron, it has a local receptive field. It doesn't actually integrate over the whole LGN, but some region of the LGN, so different, so there's like this locality assumption that's sort of known from a lot of physiology and anatomy. I think that's what I say here. The same operation is assumed to be applied earlier. So we say V1 neurons like Gabor filters. You don't mean Gabor filters here and not Gabor filters there. You mean Gabor filters there, repeat, right? So these are things that are in the textbooks of anatomy. We actually knew from work of like David Heeger and Tony Movshon and Matteo Carandini and others, things a lot about the kind of processing sort of local motifs that approximate like V1 neurons, especially where you take this kind of Hubel and Wiesel linear filter, some sort of nonlinearity, and then there's a gain control with some pooling on it. So this is kind of the canonical circuit that approximated a lot of V1 data from not only V1, but also other areas like MT with Nicole Rust and others. So we had these kind of basic ideas that I referred to earlier. We also said we don't think V1 neurons is like one type of filter. There's many types of oriented Gabors, many orientations, many spatial frequencies. So there's multiple filters at each level. And so basically this is again a summary of a lot of stuff that was known, right? And I've just kind of overviewed a lot of it. I added a few things that I mentioned to you, like timing and so forth. This is not everything, but we knew a lot. And the thing is, people were using this data way back as Fukushima built the first model in 1980, which is this kind of this neocognitron, which is a stacked series of levels, which is kind of one of the first really built deep networks that was trying to approximate this original brain deep network to say, let's see if we can get this thing to do anything interesting. Later, Tommy Poggio's lab kind of picked up this line of work with called the HMAX class of models, and this is one from Thomas Serre and this kind of this time epoch here, and that actually predicted some stuff quite well that they had fit with it, but it still was far from explaining what was going on. And my lab started doing these kind of models around 2008 to 2010. This was the kind of model class that what we looked like back then. And then in 2012, a postdoc in the lab, Dan Yeamans, I'll show you next, built this model that we called HMO, and I'm going to show you that one in a minute. But I want you to see that all of these models were following this sort of general family class here that we were sticking to because we're neurobiologists and we want to build models of what the neurobiology is doing, not because we were trying to solve computer vision. But we kind of felt like it's solving the same problem. And so there was this convergence ultimately that computer vision models ended up looking a lot more like neuroscience models much more recently. So here's this model that Dan and Ha, who is a grad student, Ha Hung, built when they were in the lab. And basically it's a set of filters applied, multiple filter types. Think of multiple Gabors, convolutions, so you apply the same filters everywhere. And you repeat these linear, nonlinear operators that I talked about with Tony Movshon's work. And these kind of repeat till you get this stack that gets deeper and narrower as you go up. So it's kind of now, this class of models is now called a deep neural network. This was just one deep network that we were building at the time. But I want to highlight that neuroscience kind of set the architectural class for us, at least constrained it pretty strongly. That's why we were working in this space of models. And so this has been going on for decades, that this has been driving the family of models towards this kind of class of models. And this is just one example of that. And then what Dan did and Ha did that was quite clever is we said, look, let's train this model to do the computer vision task. Even though we're not a computer vision lab, we said, look, we think the task that this part of the brain is solving is something that cognitive science had told us before. Look, humans, as I put in the beginning, they're very good at generalization across things like pose and position and lighting, as everything that I said earlier on, the task I introduced you to. So let's optimize this kind of architecture to solve that kind of task. Look, there's no neurons yet involved, right? We're not doing neuroscience. We're just doing forward engineering under kind of indirect neuroscience constraints here, some of them recording, some of them anatomy, and some ideas about what you think the system's doing. And so you bring those things to bear, you put it together, you're doing forward engineering, and now you're doing engineering, you're like, how am I going to optimize all the parameters of this? There are weights, there are kind of parameters in here, lots of them that you have to optimize. And here we just use basically a bunch of applied math, machine learning, computer science tricks to optimize it. We didn't consider that to be biological, but we said, let's get this network in a space of stuff that does what we want it to do under an architectural constraint class. Everybody with me so far? That's all we were trying to do at the time. Okay, so probably many of you already know this story. I don't know. So basically, the first thing we checked is this model was actually in the game, unlike other models I showed you. HMax, our own model, plus 09, this B1-like model they said was pretty good. They were getting crushed in these high variation conditions. IT did quite well, as I said earlier. This is performance. Humans were doing, similar to IT, which is not surprising what I said earlier. But this model was actually suddenly quite in the game. So that was already good. This kind of model could do what we thought we wanted it to do. And then what we did was we took this model and said, okay, this was the reason we built this model, is to say, is this a model of the brain? So we didn't optimize on any neural data. We then took the model features, locked out all the parameters, and said, can I take neurons out of V4 or IT where we had recordings and ask, does this thing, which we thought might be like V4, we actually checked all levels, but we was like, does the nearly last layer look like IT? Does the second last layer look like V4? This was kind of what we wanted to ask. And the way we do that is we ask, can we fit each IT neuron as a linear map from here to here? So we have to use some of the response data from each neuron to build a linear basis. Similarly here, we have to use some of the response data to build a linear basis from here to here. We're asking, do the neural responses live in the linear span of these feature sets built by this kind of model here? But most of the deep network parameters have already been locked down. Essentially, all of them have been locked down. Just this mapping step remained when we do this fitting. And so here's what it looks like. This is a V4 neuron. This is a response to a bunch of images. There's something like a couple hundred images shown here, just so you can see what's going on. This is response. This is not time. They're grouped by category of the object here, just because maybe they're, maybe you think they're category neurons. You can see clearly they're not. There's high responses for some things and low for others. And I'm showing you a few examples of the images here. And you can stare at these all day long and try to, as Conrad said, try to come up with tuning curves. If we use simpler stimuli, like some have done in V4, I'm sure we could have come up with a story about curvature or some other 3D thing that we wanted to come up with. I'm sure we could have done that. From our point of view, we just wanted to look at this as raw data that needs to be predicted. So these are complicated data to real-world images. Can we explain it? You can't explain it easily in human terms. I don't think tuning curves is the right way to think about it, so I completely agree with what Conrad was saying. But you take the features out of this HMO model, level 3, which was a second to last level. You do the mapping step, and you can nearly perfectly, within, not perfectly, very highly, 50% explained variance, fit this neuron here. Even though it looked very strange, it naturally can be matched by this complicated model, by its internals. And I want you to see, this is actually a better fit than this. I'll show you this in a summary mode. This is the best fit. This is the features in layer 3 trying to fit this particular neuron. This is not the best neuron. This is a typical neuron in V4. And so this is really quite remarkable that we didn't use any neural data other than to make the final mapping of the feature set to the neurons to do this. And here's an IT neuron, what I was mostly talking about. People might have more easily looked at this neuron, and they have done this. Look at this neuron and said, it's a face neuron because look, it responds mostly to faces. And I'm sure this is a face neuron by criteria of is it a face neuron or not, but it's not really a face neuron because it's structured. So it's not described by a simple tuning curve that's a step function in faces, right? It's some complicated thing that may have a lot to do with processing faces, but it's not simply a face neuron in a tuning space. Something more complicated. And you can see that's the black. The red is the fit of the model, of the top layer of the model to this particular neuron. There's some example images shown there. Here's another IT neuron. It's harder to describe. Maybe you'll call it a chair neuron, but there's some boats, maybe some edges neuron. You can stare at it if you like and come up with something. But again, the model actually fits it quite well. Here you can see R squareds now, about 0.48 there. What's that one? 0.55. The average, the median over all the population, these are just examples, is about 50%. And I want to stress that these are predictions. So we use some of the neural data to fit the features, and then these are all predictions. So the model never saw these images until it had to predict what the neuron was going to do for these images. So this is to the generalization point that Conrad made, that you actually want things to generalize, and this is generalization over images. We can also test other types of generalization by how much mapping data we give the model if you're interested in that. But this is kind of, now we have a predictive model to this degree. And again, I've already said this, so I won't say it over again. Here's a summary of the explained variance. This is the V4 fitting. You see level 3 fits on average 50% of the variance. Level 4 fits this IT data 50% of the variance. So that was to us a huge improvement over a bunch of other models that you see here as control fitting models that we had at the time. And even ideal observer controls where we kind of trained, tried to fit IT neurons with saying, oh, if you knew it was a face, you try to fit tuning functions that were like face tuning functions, that's what this would be. or chair tuning functions or any kind of tuning function in the category space. Those don't fit it nearly as well as this more complicated model that's harder to explain in human terms. I'll skip that in the interest of time. There are other ways that you can compare them, and they actually look even better than these single unit predictivity metrics. So then to kind of end this story here, what happens is we kind of took all this, we had built this model, we were impressed that it could fit our data quite well, not perfectly, but much better than previous things. And right around this time, we hadn't published this, we had that model in 2012, then this Krzyzewski work comes out, which if those of you don't know, this is AlexNet. This is the deep network that basically put deep learning on the map because it won the ImageNet Computer Vision Challenge in 2012, this particular model. And then after that, all the computer vision folks said, oh, deep networks are the way to go. And from then on, it's all deep networks, not only in vision, but as you know, in many other fields. So this was the model that kind of brought, Geoff Hinton was one of the senior author on this, Jan LeCun. Those folks that had really stuck with this idea well back from the 80s, again, back to the introduction of the talk, they stuck with it. And eventually, it kind of was the right idea. And I think now it's sort of taken over most of machine learning and a lot of kind of AI kind of work. Was this surprising? I often hear from computer vision folks, wow, I'm so surprised that those deep networks do so well. And I just always find this shocking because I'm looking at a brain that's a deep network. I'm like, some deep network has to do as well as the brain because that's what we're looking at here. So it didn't seem surprising to me, but I find it surprising that some engineers find it surprising. This particular CNN AlexNet, so this is a key point I want to make for this audience. Even though we built that network and it fit our data, we took AlexNet and we said, well, let's play the same game. They optimized it. They did the forward engineering. They didn't use the neural constraints maybe as tightly as we did, but let's just take their model and use it and see how well it predicts V4 and IT. And it turned out it predicted our data better than our own model predicted our data better. And I think that's great because it says whatever is going on here wasn't us sort of cheating or building a funny model. It was like these guys actually built the model and it fit our own data even slightly better than our own model fit the data. So the way to think about that, the way we thought about it at the time is that you have the models, as they got better within this class of these deep networks, as they got better at performance, this is a sort of set of model features shown here. Don't worry that you can't read the details. These are just different model features. Up here is that AlexNet model. This is the ability to fit IT. Again, about half of the response variance explained in 2012 by that model there. And so the summary here, and I think this is very resonant with what you heard from Conrad, is the way we think about, the way you understand the system is that you have this neuroscience-constrained architectural family that was this now called a deep convolutional neural network family that follows all those neuroscience principles that I showed you earlier. You have, secondly, some derived task, which I'll call a cognitive science-derived task, in this case, invariant categorization. And then you have engineering optimization that I mentioned earlier. In this case, there was a lot of tricks involved in how these models get optimized, large image sets, stochastic gradient descent, other engineering tricks that come into play to get it to work, some of which are biological, some may not be. For us, we just, again, ignored them and just said that's what we had to do. We most were interested in number 1 and 2. And this produces a decent approximation of what I'm calling nature's neural mechanism. It's adult mechanism, not its developmental trajectory necessarily, but its adult mechanism of producing the feature set in IT. And if you're interested in this, Dan and I talk about how these can be applied more broadly to sensory systems in this review paper a couple years ago. So here's the basic upshot, though. I don't, again, want you to think that we're done. Look, we're at 50% explained variance. So you might look at this plot and say, aha, just keep going. Make the categorization better. Just wait for computer vision to get better. And we'll figure out the brain better because it'll fit your data better because that's the trajectory that we're on. You could think of these as sort of, our model would have been here, and these are older models in a way. That's a way to think about this. But really what happened, and then if we start looking at this in 2014, actually then the curve kind of turned over a little bit. So and what was going on, people are building models that were deeper and deeper and deeper. Okay, so they didn't care about the neural architecture. They cared about performance, and they're achieving performance, but they're not guaranteed to achieve neural predictivity because that's not their goal. Their goal is to get computer vision performance. So, but our goal is to explain the brain and its mistakes and its successes and its failures and its internal predictive power on the neurons. So, in some sense, this is good. It leaves us a playing field to say, look, you can keep doing your computer vision optimization. We're going to optimize more within a space of models that are more brain-like, and that's kind of what we're trying to do now. So you can't just do performance. You have to bring other constraints in, and that's what I've tried to outline here. And so number 1 and 2 are actually the most interesting to us, although 3 is deeply important, and Conrad already referred to that. And I want to point out that it's not like these models fit. Not only do they not fit all the neural response variants, but they're not still as good at the behavioral level of either monkeys or humans. And that already came up when we saw the adversarial examples early on in the first talk. So it's known that these don't actually perform at the levels we do, even in these core recognition tasks that they put out. So again, the game is not done. It's sort of, we've gotten a lot of progress, but there's a lot more to do. And I think, do I have any time left, or should I just end? Because I could give you guys a teaser at the end. Okay, so that was all. And some of you, if you heard me talk recently, I'm sorry. I haven't told you anything probably you haven't heard before. Hopefully the rest of you, it kind of brings you up to speed of what we're doing. Maybe I'll just give you a taste of what we're doing now. And one of the things we're doing now, I don't have time to tell you everything, but this was to just give you a feel for something recent that relates to what I just told you. So this is Cohen. He's a postdoc in the lab, and I just told you that these deep CNNs are not as good as non-human primates. Here's some concrete examples of this. Each dot is an image. This is D prime performance. This is monkey. I could easily plot human. They're very, very similar, even at this image level. I said that earlier. I didn't show you. But they're very similar here. And what you want to see is this is the unity line is where the model and the monkey are matching. So images that are easy for the model are easy for the human. Images that are hard for the model are hard for the human. So that's here. But you see all these images up here where the monkey's still outperforming the model. We call those challenge images. So we basically screened a whole bunch of images, some from computer vision data sets, some from the synthetic sets that I showed you earlier. And that's all piled in here. I'm not discriminating that for you. But we were screening just to find images that were challenging. And these are up here. We call those challenge images. Call these control images because they're matched. And now we can start to look within the brain and ask about what's different. Can we see anything different going on these kind of challenge images and control images? And we can do things like do it at equal levels of human difficulty, which is a nice control for human work. So you could say everything that's equally easy for the human, like this band, I can compare this to that and say what's different in the brain, because somehow the brain's solving this, yet the deep neural networks aren't yet solving it. So that's where we're going next. And you can see this is back to what I did at the beginning, like let's find a gap between current engineered systems and what the kind of the biological system actually does. So here's kind of just to kind of outline for you what we've done so far on this. Remember, we take a deep neural network as approximating what's going on here. And without showing it to you, we can decode at an image level, at temporal resolution, we're able to measure image by image, do we see a solution to this image in IT? How good is that solution, measuring the populations of IT neurons, and at what time does it appear in IT? So I'm not going to show you all the data. Again, this is kind of hot off the presses, but I'm going to give you a feel for it. So we can measure that at high resolution because we're using those array methods that I showed you earlier. And so we measure that. And the first thing we find is that you can, even those challenging images, the answer shows up in IT. So you might have first thought, well, those challenging images, they're going to go through the dorsal stream or they're somehow funny. The answer still shows up in IT. I can't yet say that for every image, but on average, the answer shows up in IT. The interesting part is that the answer doesn't show up at the same time for the challenge images as it shows up for the control images. It shows up a little bit later, about 20 milliseconds later on average for the challenge images relative to the controls. So the solution pops out here, but it doesn't just kind of bang its way through and hit up here in like 100 milliseconds. It's like there's a lag to get the answer, but it's a very short lag. It's not like an eye movement lag. It's within the sort of inference time of the sort of what we're calling the ventral stream engine. It's within that kind of first couple 100 milliseconds of processing. And then, intriguingly, and related to this, we think, is that if you take a deep neural network model, as I just showed you, as a fitting IT neurons, when you look now closely, now that we have this temporal resolution in IT, you can ask, when did the deep network models fit the IT response? And maybe not surprising to you, but I think nice to show, is that they actually fit the response quite well at the beginning. So here's kind of what I'm showing you here. So here's the fitting of the IT neurons. Here's explained variance. Remember, we were up at about 50%. So here's the amount of IT neural response variance explained by a particular CNN model. I think this is AlexNet in this case. And this is how well you explain the IT response variance as a function of time. So you can actually explain the response variance quite well here, and then your ability to explain kind of falls off to these like 10% levels quite rapidly, as you see here. So the deep network actually is a pretty good model here, and it's a pretty bad model right here. And I'm showing you with arrows here without showing you this, that the solution to the control images kind of pops out around this time, and the challenge images, the solution pops out around this time. So you can kind of see this is all kind of consistent with the story that, hey, the deep neural networks are approximating the feed-forward kind of response edge
quite well, and that solves a lot of things, but it doesn't solve everything that this brain is able to solve. And you might be looking at this and going, hey, the neurons don't respond. For the aficionados, actually, there's actually more variance, more reliable variance to explain out here than there is out here. So this drop is not because we don't have good data or good reliability in the neurons. It's just that the models don't explain it so well. That's what this line is here. So there's nothing really to explain out here, which is why I'm not showing you anything. But now we have response variance, and we still can't explain it as well here than here. So you guys already have ideas of what this means. We have our own ideas of what this means. And our preferred idea, and it's been around for a while, is I kind of told you there's these feedback connections and these recurrent connections. And we don't know if this is recurrence or feedback. But the ventral stream is not a straight feed-forward system. Even though we approximated it that way as a sort of stepping stone, we've known all along it's not just a feed-forward system. So now we think we're ready to start engaging in, if we have recurrence in here in an interesting way, can we better predict models with recurrence? Can they better predict the responses up here? And can they perform better on this kind of challenge images? So that's what we're trying to do now, is building models within that class and see if they can better fit things up here. And we're trying to develop methods that we can silence the connections, the feedback connections specifically. This is done with retrograde virus tools. If we can do that in a monkey, then we can test whether cutting those connections in a time-delimited manner will produce deficits in behavior and changes in responses that are consistent. That's what we're trying to do in the lab, but that's very early days on that. So that's kind of where we're headed there, and this is just a teaser rather than a full story. None of this is really published. It's been at cosigned, but you're sort of seeing the latest edge. And I don't know, I put this up here after watching Marissa's talk that maybe I can think of this kind of evoked but slightly later thing as being part of an exogenous attention network. I don't know, that's a point for discussion. But something automatically is evoked. And this happens, I should say, by the way, even if the animal is doing a task or not doing a task, the solution still pops out and the lagged solution still pops out, regardless of whether the animal's active in the task or just passively viewing the images. Okay, so just to summarize what I told you here today, just in case I've gone too fast, I told you that I can take ITs and make a pretty good approximation of behavior. So that's a linking hypothesis from IT population vectors to behavior. We had some questions about that. I then told you that encoding deep network models are very good approximations, about 50% of the response variance of IT and V4, these kind of deep network encoding models, but still not done yet. And then in the last, I kind of teased you with some data to say there might be something about recurrence or feedback that is the next frontier for us to figure out here, but I haven't fully answered that for you. So that's kind of where we are, and I want to end by thanking these are the folks that do the work. I hopefully thank the main ones along the way, especially Dan Yeamans, who did a lot of that deep network modeling. He has a lab at Stanford. And all these people have done a ton of work, and they deserve all the credit. I just get to present and talk about it. So thank you for listening, and I can have some questions.
Yeah, I mean, that's kind of, I think Conrad would probably agree with that. I think that's the frontier now for us. What we think about the space to play in is the space of objective functions. That's why I gave you those 1, 2, and 3. It's like, what's the architecture? That's where neuroscience should kind of try to bring what it can. What's what we call the task space or the cost space? It's not necessarily pick the right one. There's probably a layered sequence of objective functions where the dorsal stream drives the ventral stream as an implicit supervisor, right? These are the kind of probably the ways to think about how the system builds itself up from development. And that's this. Now you want to build models with different kind of task costs on them. And for us, that's what we're trying to do. And I think others are in that same game, I hope. But for us, we don't care as much about the optimizers, one game we're playing now. It's like we're not trying to worry so much about, as long as we can get an answer that we're hoping that it doesn't matter much how we optimize. But that's just because of convenience that we don't, it's easiest to optimize using the sort of standard tools. Well, that simple way of thinking about it is, I told you there was a good IT feature set, but I didn't tell you how it got there. And I think it gets there largely in an externally unsupervised way. And what I was trying to separate, and it's not going to be this clean, but the sort of simple model is like externally unsupervised developmental scheme gets you IT ventral stream features, followed by reinforcement supervised scheme. For us, that was the monkey getting some juice when he did his thing. For each of us, it might have been our mom saying, when you said dog, you're like, yay, right? So you get a few labeled examples that's supervised, that's going to actually build the final step there. And then you would say, well, look, given all that, the power of the system is in the feature build. How did you get those features? And so that is kind of the deepest question, is how do you develop those features? Probably by an internally supervised, looks externally unsupervised system to build up the ventral stream. And that's my guess, and now it's a matter of building forward models like that. We're not the only ones sort of thinking that way. What I hope we can do from a neuroscience perspective is also get some develop data from earlier and younger animals to follow the developmental trajectory. Because the other alternative would have been everything about development, all the kind of getting the feature set is built into the genes, right? That the learning of the network is actually evolutionary and then somehow encoded in the genes. Our preference is that the architecture is loosely in the genes, but that the weight functions on the network are developed. But that's approximation, probably not exactly that way. But these are, to me, the key questions at the moment. It's a great question. Does that help? Did I answer? Yeah. I mean, that might matter, the binocular vision, but people without binocular vision, I think they're going to do just fine on these tasks. And remember, the monkeys are being tested. This is a flat screen, so there is no binocular cues, but you might have said during development it was important. It's hard to say. My guess would be that doesn't matter, but that's mostly based on knowledge of people with lacking good binocular vision, actually. Unless you put them in special situations, they actually are mostly okay. So I think they'd still beat deep networks on these kind of images, but I haven't tested that. Maybe somebody has or will. I don't know. That's the best I can say about that. I don't. Right. You're kind of. This is a great question. This is something we're working on a lot because what you're asking is like we're basically proposing a metric that says is this model like in the game, right? And I gave you a predictive answer. And what you're pushing back on, like if I could, you could maybe turn that into the rather than ImageNet competition, it could be IT competition, and that's kind of what Dreamin's like. But what you're pointing out, I think that I worry about too. If you just chase that metric, you might go the wrong way because you could end up building models that have like very wide reservoirs of features that can still predict. And we don't think that's exactly true in these models, but I think it is, the way I think about that is like you can check dimensionality of both. That's one of the ways we're looking at. The other is you can ask the neurons to fit the models. And if the neurons can't fit, you got to make the predictions both ways. And that's what we're trying to do in the metric space, to have a more complete metric for exactly the reason that you say. If you really want to say they're matched, you need it. You can't have like features hanging out in the model that are not actually in the brain, right? They need to kind of match in that way. And I haven't shown you that in a convincing way. And I'm not fully convinced myself. I mean, I'm not claiming that these models are copies. I'm just saying, look, they're really good relative to what we had before. But that's the example of exactly one of the axes that I worry about. So very, very perceptive of you. Yeah. Does everybody else get that idea, right? I mean, if you have many more dimensions sort of in the feature set, that maybe you kind of, your predictive map that I was showing you could be leveraging those dimensions. And it says that you have a basis set that can span the space that the IT neurons lives in. But if you're saying that basis set is like the IT feature set, it's not you want a fuller comparison than that, right? It's like this has 100 dimensions and IT has 2. They're not the same. This one has 100 and this one has 2, but this can fit that, right? So that's why I was suggesting the inverse prediction is one way to go. But some clever folks in the audience may be able to tell me a better way to think about that. Does that make sense? Is it sort of? I don't know. Right. The way we have tested that, so we have like face versus face tests, and we tested that. And when you build IT neuron population reads to do face versus face, train them in the same way, Joe versus Sam. You find that you get a performance level. It's pretty low. You ask humans to do the same task. It's equally low. So we may think that humans are good at those things, but they're actually not. But those are under regimes of high variation. Think about 2 faces without hair under high variation. Those are the conditions that we tested. And then the humans in the models, you could say they're equally bad. So I think a fair way to think about your question is like, well, let's put it in a space where I'm like, frontal view and a friend that I know, but for the monkey, but you want to be someone you didn't know, right? So, because you don't want training, but like, it might be. I mean, you're kind of proposing a kind of a challenge domain that we could test. And I think the spirit of extending, again, I don't want to imply that we've done everything. That is another interesting challenge domain beyond the one that we've been. Yeah, I think, again, there's an infinite number of image tests we could do. And that's a good one. I mean, we have tested these on natural images, so it isn't about the synthetic. I mean, maybe you're saying, oh, the synthetic images are somehow special, but we also have this on kind of natural image sets. You guys should know that natural image sets have their own downsides, right? That's how computer vision got into trouble at the beginning, is because like there are lots of ways to cheat. That's why we built that set, because I looked at it and go, oh, it's obviously a car, it's a car, it's a car. But the computer vision sets at the time that were so good, it's like, what? What is that, right? Because they were cheating on the background, which they could no longer cheat on. Right, so that's why we originally did that set. And then, there was a long time, like they moved on, but we still were busy collecting our neural data. But you're exactly right. I mean, testing these things in richer domains, these faces, we think about time, like movie stimuli. There's lots of ways, to extend the difficulty of the challenge. And I hope that I've given you guys a framework of kind of, look, you try to have a decoding model, you see how well it fits, it should generalize over something, and how big is a generalization domain is the question you're asking now. And if we, as a field, keep asking those questions, we'll get to, I'd like to think we'll get to understanding, as Conrad was implying, right? But you're proposing a particular type of generalization. That's a good one. It just, we haven't done it maybe the way you'd like to yet of that. So to be continued, I hope.